Good morning, everybody. It is May 11th, and today is going to be our last day talking about DNA, right? And so before we get into the PowerPoint, just a quick little recap, all right? DNA is the genetic material inside all living things. And that DNA is made up of nucleotides, all right? And those first four nucleotides that we learned were adenine, thymine, cytosine, and guanine, right? Those are nucleotides found in DNA, right? And so the first time that we took a DNA strand and really broke it down, we were doing DNA replication. And that was just making a DNA to DNA complementary base pair, right? And that is the type of DNA replication that cells undergo before they go through mitosis, right? And the lecture after that, we went into RNA transcription. And that is taking a DNA strand and making an RNA strand complement, right? And what happens in RNA is that thymine is replaced with uracil, right? But besides that one thymine to uracil base pair switch, the process of finding the RNA strand complement to that original DNA strand is very much like replication. But again, there's no thymine in RNA. There is uracil. All right. And so today we are taking that RNA strand produced through transcription and translating it to a protein sequence. All right. So translation is taking something that is a genetic code, all right, RNA, and it's taking that genetic code and turning it into a protein sequence. So going from genetic base to protein, that is translation. All right. So let me start presenting my screen, right? And we will go through the PowerPoint, and then I will explain the assignment attached to today as well, all right? And so going through some vocab first, all right? We are doing protein synthesis or translation. All right, so protein synthesis, you're making a protein. Translation, again, you're going from genetic material to proteins. All right, introns and exons. All right, I spoke about this a little bit last lecture, but introns are those coding regions of your DNA that are eventually cut out because they don't code for anything important. All right, so when we're trying to make something derived from that DNA, we want to make sure all of that quote unquote junk DNA is cut out. All right. And so exons are the parts of the DNA that we want, right? And splicing is the process of cutting out those intron areas that have been reflected in the RNA complement, right? And polyadenylation, all right? So we have the nucleotide adenosine, all right? And so essentially just as a big stop sign to the eventual ribosome reading this piece of RNA, a poly-A tail or a polyadenylated tail tells the ribosome that the RNA strand is ending, right? And so once it starts going from like GCU, GGCCC, and then it goes A, it's just reading a bunch of A's. It knows that the sequence is pretty much done, and it stops reading that piece of RNA. All right. And so steps of translation, right? So it has to go through splicing first, all right? So that RNA strand that just got made based off of the DNA complement, all right, that RNA strand still has the complements to those intron areas that you don't want reflected in your protein sequence that's going to be made through translation. All right, so we got to splice out those intron areas, all right? Some editing goes on. Essentially, you have like enzymes that will read your RNA strands and make sure that they are correctly sequenced because, again, you're about to make a protein and you don't want to make the wrong protein, all right? And that poly-A tail is just the stop sign at the end of the RNA strand, all right? And this is just a refresher. So adenine, thymine, cytosine, and guanine exist in DNA. And then once you go to RNA, all right, that thymine is replaced with uracil, all right? So mRNA is going to leave the nucleus, right? It finds a ribosome, right? We learned when we were going through cell structures and functions that ribosomes are the machines that make your proteins. And so the ribosome comes out of the control center, out of the nucleus, and then it finds a ribosome, right? And then that ribosome just clamps over this RNA strand and reads it a codon at a time. And again, a codon is a triplet of 3 nucleotides. So GCA, that is a codon: guanine, cytosine, adenosine. All right. We're going to start going into codons a little bit later. The ribosomes read the sequence, yep, and then molecules of the transfer RNA, all right, they'll bring those amino acids to the ribosome, and then they will make a sulfate bond, right? And again, because we are trying to mix in a lot of learnings into one video a week, I'm going to have to not explain some of the things, but you'll notice that mRNA and tRNA are mentioned in the slide. Messenger RNA is read by the ribosome, right? And the tRNA are these little free-floating pieces of transfer RNA that are going to transfer amino acids in suspension in the cytosol to the RNA that needs the eventual amino acid carried by the tRNA. All right, and this is what the tRNAs look like. All right, and so that mRNA is on the bottom. That's that 5' to 3' strip. All right, and so this tRNA is going to as the ribosome is reading that codon, that GCC on the ribosome, the tRNA is actually considered an anticodon. Codons and anticodons will bind, all right? And it's looking to pair up this GCC with a CGG, all right? That's how it knows that it's the correct amino acid. But you'll look at the very top of this organic structure, that the amino acid is in that top dashed green box after the red portions, right? So it's like a little carrier. All right, the anticodon is going to find that codon complement, and they are going to bind, all right? And eventually, as the amino acid bonds continue and the process continues, it's going to reach that poly A tail at the end of that RNA strand, and it is going to stop producing more of that protein, right? And so there are 3 videos in this PowerPoint that I am going to let you go and investigate on yourself, all right? So that's pretty much the end of the PowerPoint, all right? So I want to explain a little bit more offhand, and so I'm going to stop sharing my screen, all right? And so the beauty of genetics is that the adenosine, thymine, cytosine, and guanine, those base pairs are only 4 building blocks, but those 4 building blocks arranged in different sets and series and such and expressed differently is how life gets different. Right, so the adenine, cytosine, thymine, and guanine found in us as humans are the same things that are found in plants, in dogs, cats, you know, cows, horses. You know, any type of organic life is going to have genetic material, and that genetic material across all species, across all life, is mainly made up of the same building blocks. And so in biochemistry, when we were learning about what nucleotides and the monomer of genetic material was, there was a nice stepping stone. And now that we have gone through genetics, which is essentially how parents give their genes to offspring through you know, meiosis and such, but we didn't really know how those jumps and leaps came from the parent to the offspring. It's not like they sneezed and then they're just like, yep, there they are. That's my genome. It takes a lot to really take a magnifying glass and pretty much just specify down to the genetic level of just base pairs and such. But we have 3 billion nucleotides in our genome, and your genome is encased in every single cell in your body, and we have trillions of cells in our bodies. And so there is an exponential amount of genetic material inside all of us, and it's just kind of humbling to think how much can fit into something so small, right? And so at the end of this video, we should be able to take a DNA strand, find a DNA complement, right? That's DNA to DNA, right? We did that in the first video. And then last week, we took that DNA strand and then found the RNA complements to that. And that is, again, switching that thymine into uracil, right? And this week, I just went through a little bit of RNA translation, right, which is going to be reading that RNA strand and finding the amino acid sequence, all right? Today's or this week's homework is going to be taking a DNA strand of just sets of letters, A's, T's, C's, and G's, finding the RNA strand, and then finding the amino acid related sequence. All right. And so I'm going to stop this video because this is a lecture video, and then I'm going to begin an additional video to explain the assignment. All right. So thanks so much for watching. Keep up with the good work, guys, and keep an eye on the grade book. All right. I'm updating that as frequently as I can. Awesome. All right. Thank you, guys.